From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 8, recorded on July 6th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York City, Ori Lieberman.
Hi, Vincent. How are you doing?
Okay, how are you?
Are you getting the storm that's passing through?
It's making threats, but hasn't done much yet. No.
I woke up a bit ago to thunder and right outside. Like the lightning must have hit right outside the window. It was crazy.
Yeah, when that happens, it's scary.
Yeah.
I saw you on the sidewalk the other day, right?
Yeah. That was the first day back in clinic, so it was very disorienting.
So everybody's got masks, so I'm like, it kind of looks like Ori's upper part of his face, but I'm not sure. So I just clapped him on the back and kept walking. I said, what if it's not him? The guy's probably going to think I'm a jerk, but it was you. Okay, good. Also joining us from Nashville, Tennessee, Erin Calipari.
Hello. How are you guys doing?
All right.
Yeah, the mask thing, I've been spending most of my time realizing I'm smiling at people, but no one can see my face. So then I'm really trying to smile with my eyes. And
your eyes smile. Yeah, when you
I'm
smile, your eyes tend to smile too.
I'm I hope people understand because I keep opening doors for people and smiling. And then I'm like, all right, well, we'll see how this goes.
And from Salt Lake City, Jason Shepard.
Hi, Vincent. How are you?
How's Salt Lake? You still getting snow?
No, we have summer back. It's actually real summer in the 90s now.
Nice.
Nice and sunny.
Yeah, Nels and his wife had a kid, right? You know that.
I saw that.
Yeah.
Yeah.
So actually, this is funny. Two of my hosts on different podcasts are having kids.
Oh.
Nels had his, and Stephanie Lang, I'm not sure she did yet, but she's any day now. Isn't that funny? I don't know what it means.
Well,
I'm not pregnant. So.
I'm far away from it.
Me either. I like when other people have kids. It's always great. I get to look at them. I'm like, they're so cute. And then when they start crying, they go back.
I'm finished. We raised 3 kids. I'm done. No more. Not that.
I feel like that's a good number. I am impressed.
It's very hard, and I just don't think I did a good job, but
I'm sure you did. I don't know how the parents during this whole thing are doing it. I'm struggling just like me and a couple cats to figure it out. And there are people who have kids at home and are still being productive in lab. And it's really amazing.
Our older son is living on his own and working. He's fine. But the other 2 are out of college and they can't get jobs. So they're at home. And I'm like, when are you going to leave? I don't want you here. They bring people over. I mean... it's my house. Go. Is that unreasonable to want your kids to go off and live their own life? I think not, right?
No. I think you earned that at this point.
Anyway, I'll hand it over to you, Ori, and you have a few things to talk about and then tell us about the papers.
Yeah, I just will comment on the visa situation. So I guess a couple weeks ago now, the Trump administration announced that they are suspending new H-1B and J-1 visas, I believe. So these are visas for people who either come to the U.S. to train from other countries or who are given jobs, essentially jobs that the employer certifies cannot be filled by an American. And you guys can give me your experience from your labs, but in the labs that I've been in, I would say 25 to 50% of the lab have been foreigners. And really, I was trained very closely by a Swede and an Italian who were here on H-1B visas. So, it would be very difficult to do science without those, without immigrants. So I think it's really going to be a problem for the long term.
It's totally absurd. I was on an F1 visa, a J1 visa. I would not have been able to be in this country if I wasn't able to be on those visas. So but even back then when I was on those visas, every time I went out of the country, I had anxiety about coming back in. And they're a bit, and I'm from New Zealand. I'm a white man, and even I felt that I was being treated like a criminal coming back. And now it's exponentially worse. It's just, I feel for all the immigrants in this country. And then adding this sort of useless piece of legislation that really has no effect, no good effect on the so-called loss of jobs because of COVID. I mean, really, it's just a total political stunt to pander to a base that is anti-immigrant. And as you just said, most of the innovative science and technology that happens in the States absolutely requires international workers and students and trainees. Without them, it all crashes down, and so it's actually harmful for this country in general. It's just totally crazy.
Yeah, I think the problem is that, the idea for people who aren't in science is that, this will open up jobs for other people, but that's not who's filling these jobs, right? If you get rid of these people that are heads of labs, you're not just going to have someone step in and fill this position. Like, it's not like those aren't like they're just there and it's like, oh no, Americans are losing out. These are things that are enriching the American workforce. I mean, you're thinking about people running a lab. I mean, how many, if you want to think about it from the US perspective, I mean, how many Americans are these people training to go out and make our science better? So you have that aspect, but then you have the people that are working in labs like mine, who are actually working with lots of different Americans, enriching that environment and also producing really valuable science that then goes on to kind of support the American economy as well. And so, like, from a human perspective, this is horrible. And then also from an economic perspective, it's horrible. So it's not like there's no real justification for this other than saying we don't want immigrants here, which is ridiculous because this country was built on immigrants. I mean, my family, my great grandmother didn't speak English, came here, and it's crazy because it's unheard of that, like, I am a PhD. Like, no one in my family had, my father's generation, they were the first people to even go to college in our family. And so these kind of like things, people forget that that's like a core of what makes this country great, right? Someone can come over, you have this kind of foundation, and then you have people who can go on. And now we're trying to say, oh, well, we don't want immigrants, even though most of the people here are immigrants. It's really amazing and disappointing, I think. And people don't understand how widespread these kind of policies are.
Every lab I've ever been in had amazing people from other countries. I remember as a PhD student, there was a guy from Germany, there was a guy from Japan, and they would take me to their restaurants, to introduce me to their culture. They wanted to show me their food. I remember the Japanese guy brought me to have a thing where they cook it in a pan, and I didn't know, I knew nothing. I was this dumb guy from Paterson, New Jersey, and it would just open my eyes. But also scientifically, it's so enriching. In David Baltimore's lab, same thing. And when I had mine, I had a few people from other places. It's just the way it is. It works great. And they go back sometimes and they bring great science back to their country, right, and propagate it. So, I mean, just think of all the students from China and postdocs that we trained for so many years, and now they have their own great science establishment. Remember, they used to come here and get PhDs and do postdocs. It was just amazing. So I think it's crazy. And Ori, you said some other flack coming out of this as well, right?
No, I think that was the main thing. Oh, I guess Jason was saying the online.
That I mean, it's not clear what's happening there, but ICE just came out with this absurd statement saying that if a university has online courses only, the international students that are at that university have to go home.
It's crazy.
Let on the fact that these days, some countries you can't even travel back home. Like if I wanted to go back to New Zealand, I would have to stay in a hotel for 2 weeks as a quarantine.
Yeah.
And so logistically, for many people to go back to their countries right now, borders are closed. There's no airlines taking people. So it's just, it's one cruelty after another. It's just.
Well, then you talk about some places, right? You go home and like maybe you don't have access to the internet and computers and things that are working. Like when you're talking about online courses, people think this is a really easy thing to implement if you're not in academics. Online coursework and not having bias introduced into this kind of training is really important. Think about like my grandparents live in rural Missouri. They have like dial-up internet still. Like I couldn't go and do like a Zoom lecture from their house. Like if that is where people are going, and I mean, this is in the United States, they're going to a place that has like, it's not like it's a third world country. This is Missouri. And you can't, they wouldn't be able to do a Zoom course. And so, you're thinking about this too, is like sending people back. You don't really know where you're sending them. You're also talking about hours of, I mean, think about, okay, you're in Australia, you're talking a 12-hour difference. You're in the middle of the night trying to do these Zoom courses. It's not as easy as people are acting like it is. And I think it's going to be a big detriment to these students who, for what?
Yeah. And in the middle of an already difficult time.
Right.
So it's just like, it's just one thing after another. And this is just, to me, pure cruelty. Yeah.
So Jason, anyway. We get quite a few emails on TWiV from New Zealand. Got a bunch of fans.
Oh, yeah?
And they've been...
Are they gloating?
Yeah, they're gloating because they said, we have no more cases and nobody can come here. I think I told you that last time. They opened restaurants and the prime minister couldn't get a table. They were too crowded.
Right. Yeah, so for me, thankful that my family's safe and I don't have to worry about them. But if the things get worse in the States, I'm just going to go home, I think, and just take that 2-week quarantine in a hotel and work.
They have good internet there?
Good internet there, so, All
right.
Are you guys back up in lab?
Kind of. I mean, it's still the same. We can only have 2 or 3 people in the main lab space at a time. So we have to do rotations, but the numbers here are getting worse, and so we're a little worried. It doesn't look like it's going to get better anytime soon. That's for sure.
Yeah, we're like 50%. I mean, 50% space capacity, but again, we're in the South, so we have more space than people. So 50% space capacity is like not a ton of people coming in, but there's lots of space, face masks, the whole nine. I mean, again, like the South it's way easier to commute to work without interacting with people. So like, you walk, you drive. There's not like New York City streets and things like that make it an extra level of, danger, but we'll see. Our cases are also going up. We'll see what ends up happening. Nashville rolled back their phased reopening, so we went back a phase. So, we're a tourist town. So the problem is when you open bars, it's not the locals that are going to the bars. It's people from who knows where, and it's no masks. And so we'll see. I have a feeling it's going to continue getting worse from what I've seen over the last 2 weeks from my car driving down the street and seeing the stuff, but we'll see.
Yeah, I only have one person in my lab, and she's been working just continuously. I go in twice a week now because I don't need to be there every day. And I think it's a little bit of a risk to go in the city every day. And so I'm just going to take it easy possibly for the rest of the year, because I think New York cases are going to go up again. They're starting to go up in New Jersey. As people come in from other states, they bring virus with them, and I think it's inevitable because not enough people are immune. So.
Yeah. So we have 2 papers to discuss today, and Jason and Aaron are going to lead these. So maybe I'll hand it off to you, Aaron.
Yeah. So what we'll do, and we were just talking about this, what we'll do is we'll go through that kind of, I think that this paper that I'm going to go through and we'll go through it rather quickly because I think it kind of sets the stage maybe for some of the stuff that we're kind of excited about in the paper Jason's going over. And so this paper is Glia-to-neuron conversion by CRISPR alleviates symptoms of neurological disease in mice. And so this is actually a kind of cool framework for trying to figure out ways to treat neurological disease symptoms, especially those that are kind of punctuated by a loss of different cell populations in the brain. And so I think that, when you're thinking about these neurological disorders or neurodegenerative disorders, one of the really hard things, and I'm in the dopamine field, so I always go to things like Parkinson's. One of the hard things is when you lose cells in the brain, how do we treat this, right? Because if you have cells there, you can say, okay, we can increase or decrease neurotransmitters, we can block receptors. But if the cells are gone, what do you do to solve these kind of problems? And so I think that what we're talking about today is actually a bunch of kind of cool approaches that are changing certain cell types like glial cells into the brain into other functional cell types to kind of regenerate cell populations that are leading to these symptoms. And so this paper is actually really interesting. So it's kind of funny because 2/3 of the paper, maybe more, focuses on retinal degeneration, and then it kind of pivots and goes into kind of Parkinsonian models. And so what I think we'll do is we'll go through kind of the framework for kind of how they're solving these problems and what they're doing in the retinal side. And then we can kind of pivot over and let Jason take the Parkinson stuff with some of these other papers so that we can kind of talk about both. But essentially what they did here, which actually is kind of one of the cool things of CRISPR. So for people who don't know, CRISPR is this kind of, I think maybe everyone who's interested in science knows a thing about CRISPR. I feel like this has been like the next frontier of the brain. But essentially what CRISPR is this enzyme. And what we can do is we can use guide RNAs to kind of bring it to these places in the genome that we want it to kind of remove. So what we can do is we can go through and remove proteins, we can mutate proteins, and it gives us this kind of dynamic ability to do this in a really fast way in even living cells. So what they're doing here is they're focusing on using CRISPR to actually knock down this protein called, I always it's funny because I always know the abbreviations, but not the actual names. Polypyrimidine tract binding protein 1, or PTBP1, which I'll probably misstate like 10 times. And so what's really interesting about this protein is when they knock it out of glial cells, they can actually make these glial cells differentiate into different cell types. And so the kind of thing about this paper is they're using this protein knockdown approach they start in the retina, but they also do this in the striatum to make glial cells differentiate into other cell types that can kind of reverse neurological symptoms. So,
and this idea that you could do this had been known before, right?
Yes. Yeah. And so, but the big thing here is that they're doing this in vivo. So a lot of the stuff before was cell culture. So we said, okay, well, we can make stem cells, we can make them differentiate into other cells in dishes. And this was always like a kind of cool thing we could do. And now they're doing this in an in vivo model system, which is kind of the big thing, because the next step for taking this into a treatment would be able to be doing it in an in vivo system, not just kind of cells in a dish, because they're just more complicated.
But I would say, the surprise here is still that it's only one protein that you're down regulating, and that sets off a whole cascade.
Yeah.
Right.
And the fact that, and we'll get into this more in the second paper as well, is that there seems to be this latent program that allows the astrocyte to become a neuron. And that's the default. So that wouldn't happen if it wasn't for the case that you had this sort of master regulator. But I find it amazing that this one manipulation has this dramatic effect.
So does this imply that astrocytes and neurons are quite similar, or does this just make them very different?
So during development, there's these neuroglia cells that can become either or. So we knew that that there was always the capacity for these cells, and they are quite similar, and that the gene program in each of them is fairly similar. But once they became differentiated, that's when we thought, well, you can't really undifferentiate these cells to become another cell, because that's not a stem cell. This is a mature differentiated cell, and so I think that's the big surprise here, is that you can reverse this inside a brain.
Right, with a single protein knockout.
Manipulation.
Right.
I have to tell you that this protein, PTB, I've known about this for 2 decades because it's needed for poliovirus replication in cells. If you knock it down, the virus doesn't work. And, poliovirus, when it gets in the brain, will reproduce in neurons, and it'll reproduce in astrocytes as well, and it kills them. So I'm thinking, people who have post-polio, they're losing neurons. They had polio years before. Maybe you could give them some back, by doing this. That would be cool for any neurological disease.
Right. I think the thing about what they're doing here with like the retinal degeneration stuff is something that's close. Again, like it's really hard to take these preclinical mouse models into like a human model. But when you have like a targeted problem, like, okay, the eye, we are missing cells in the eye. It's not easy, but it's easier to use, like kind of thinking about using a viral approach to say, okay, well, we'll inject this into the eye and then that will fix this. When you have things that are kind of whole body, it becomes a lot more difficult to say, okay, well, if you identify where it matters, maybe that's maybe then you can reverse it there. But I think that's the thing with CRISPR that maybe isn't always relayed to people who aren't in the science field. That's like, oh, we'll just CRISPR it and it'll work. But you can't just like give CRISPR to somebody. So
it's the bottleneck is delivering the CRISPR in the first place with gene therapy. And the eyes are one of the few places that's actually really been demonstrated in a human that you can inject the virus. So that's, it's pretty cool.
Right. Well, and I think that's why I was excited about this kind of this component of this. So essentially, I guess we can kind of go through the first thing they did in the first figures is just to show that they can actually, knock down this PTB protein. So they generated a bunch of different kind of guide RNAs for different parts of the gene. And what they did is they went through and said, okay, well, for these different guides, how much does it knock it down? And they found this guide RNA that's at between exons 5 and 6 that knocks the protein down like 80%. And so that's, they were like, this is great, we can do this. And then they also actually, interestingly, show that they can knock it down in astrocytes and neuro2A cells, which are kind of neurons, and did RNA sequencing to show it was knocked down. I was a little surprised by these data showing that like the only thing changed was the protein they knocked down. I guess it's because of the time point. But it just seemed like an RNA binding protein knocking it out would be like have wide scale functional changes across the transcriptome.
Yeah. And in fact,
that's the implication of the rest of the paper is that knocking this out should have massive changes in the transcriptome. So it was a little bit strange.
That's my first thought. I was like, why is it only knocking it down? And I think their argument is all the other stuff was kind of longer time points. But even so, transcriptional regulation, we're talking like minutes, like we're not talking days. And so this was like 2 days and it's still only this protein, which I thought was kind of interesting. And it's pretty, like nothing else looks like it's really changed. So I thought that was kind of an interesting thing that was, I don't know what could explain why that happens, but
I also would have loved to see this done in vivo. I mean, we've talked about single cell sequencing before in other contexts, but this would have been really one of the like ideal uses for this, where they could have purified or selected for cells that were expressing the guide RNA and then done single cell sequencing and kind of tried to see developmental stages of these cells after knockdown. But they do this in culture.
And then they do this in the next paper. So, it's doable.
Well, that's why I think it's good to go through this rather quickly, and we could talk about exactly how they're doing it in the next paper, because I think the next paper is like the next step from some of these. In the next, so then essentially what they do is they take this approach, they validate it, and now what they want to do is they want to go into kind of these animal systems and see how manipulating this protein in glial cells is going to change the distribution of cell populations in the retina. And so what they do is they have these like multiviral systems with transgenic animals. So what they're doing is they're expressing Cre recombinase and GFP on a GFAP, which is a glial cell specific promoter. And then they're also injecting a virus containing their CRISPR constructs now into these cells. And so what they're doing is they're only knocking down this protein, this PTB protein, in glial cells. And then the question is, when they do that, when they start looking at these cells later, are they only expressing glial markers or are they expressing markers of other cell types? And so what's kind of cool is they show that essentially when they knock out this protein in vivo in glial cells, these cells that are expressing the fluorophore, so they were definitely glial cells before, and now expressing the markers of some of these retinal ganglion cells. And so essentially they did this a bunch of different ways. So this is like the one in the paper with the specific, they did it with like non-specific promoters. And so what they're showing is that these cells are starting to see markers of these cell populations that are no longer glial cells, that are now markers of these retinal ganglion cells. Which was a kind of neat thing, right? You can just do this. And this is in like a normal awake animal, and this is just a normal retina, and so that's what they're seeing. And then they went through, so then the next thing they do is they go through and they say, okay, well, that's great, but what happens if we actually do this in a model of a degeneration? So what they do, and so this is in figure 3, they have this mouse or this model where they inject NMDA into the retina, and what that does is it kills all of these cells. So they show that they can inject NMDA. Now you're starting to see that these retinal ganglion cells are basically gone. They're dead. They're totally gone. And then what they do is they do this viral approach again now, where they're doing the same kind of thing they were doing, where they're using this CRISPR construct to knock out this protein in these glial cells. And the question is, can they kind of restore the distribution of these retinal ganglion cells that were now removed with this approach? And so what's actually kind of cool is that you can see that they're totally basically gone, but then after they do this approach, you start to see that there are more cells expressing these markers that are markers of these retinal ganglion cells. So BRN3A, RBPM. And so these basically, you're seeing this increase. It's not like a full restoration. You're not like at 100%, but you're seeing a significant increase in this of these cells in a degeneration model, which is kind of cool, right? Because that suggests that maybe you can start to use these approaches to restore, even if it's partial restoration of maybe function. So I think this is my big thing for this paper is going to be, do they show that they're actually restoring vision, or do they just show that they can restore light sensitivity, which are different things? Well, I mean, again, first step, right? Can we restore light sensitivity? The next step is talking about organization and whether that light sensitivity can be translated into some sort of actual visual like system. And they kind of start at the first step. So they do that. They basically patched onto these cells to record their activity, and they show these cells that they're actually differentiating into these retinal ganglion cells now. They respond to light. So they have that LED. So they have some that are respond when the LED turns on. They have some that respond when the LED turns off. A majority of them respond when the LED turns on. So this is obviously possibly they didn't patch that many cells, but it suggests that they might be biasing towards one type of cell population, which is good or bad depending on what you're trying to do. But they also show that they're functional in some way. So they are responding to light. So you've replaced cells, you've differentiated glial cells into retinal ganglion cells that also respond to light. So that's actually a kind of cool thing in a neurodegeneration model. So the other thing too, so this is actually kind of cool, right? Because the fact that they respond to light is pretty neat. But the next thing is do they integrate into the actual visual system, right? So you have the response to light in the eye, but is that actually getting transmitted to the pathways in the brain that are actually encoding this information? And so they did a bunch of kind of cool studies where they were just looking at just like where like these cells were going. And so they started by saying, look, they are seeing the expression in the optic nerve. So they're seeing that these cells are actually kind of starting to integrate into the visual system. They saw it in the lateral geniculate nucleus and the superior colliculus. So they're seeing expression of these cell populations there as well. And actually what I thought was the kind of cool study is they recorded in the visual cortex, and they flashed a light stimulus in front of the eyes that they had done these lesions in. And what they found is when they did this CRISPR approach, that they basically restored visual cortex responses to these lights. This light. So if you look in figure 4 on panel G, you can see that the light stimulus, and the stimulus is just a flashing light. So you can see that in the normal and the wild type, they have these responses. When they lesion these cells with NMDA, they go away. And then you see some recovery of these responses in the visual cortex when you actually do this CRISPR approach in these glial cells. So not only are they responding to light, they're actually integrating into the visual system as well, which is kind of maybe the next step.
To me, this is one of the most incredible points because a lot of times the way I think about axon pathfinding and kind of regeneration is that it's limited by developmental time.
Right.
And in this case, right now we have an adult animal where there are axons projecting from the retina to the thalamus, to the lateral geniculate nucleus in the adult mouse. And it's crazy. And I think that this is, it is a functional response. I think that in the next paper, they really get at kind of the mechanism of this functional response. But one explanation for the increase in visually evoked potentials is that there are new axons growing from the retina through this pathway.
Yeah.
Which is awesome, right? Because it's not maybe as hard to get a cell to respond to light. The question is, does that cell integrate into the existing system so that light response means something.
Yeah, and we actually my lab does VEP recordings, and actually the VEP recordings in the wild type mouse and the untreated mice look weird, but the ones in the recovered mice look better than I actually would have liked to see a little more quantification here of what these VEPs are looking like, but it is astounding because one of the big drawbacks of when stem cells were sort of touted a decade ago as being able to regenerate in any system, we soon found out in the brain that if you stick a bunch of random stem cells, they don't integrate properly into the circuitry for many reasons. And so it's kind of been a big bust because we haven't really, neuroscience haven't found a good way of directing those stem cells to integrate properly. And here you have this one manipulation that's basically converting an existing cell in the system to some other cell, yet it seems to be able to integrate much better than a stem cell. So
right. And my question as a processing person is going to be kind of, what does this integration mean to the animal? So they did one thing that I think is interesting, but maybe doesn't get at what I'm thinking. So the next study, what they did is the first thing is, okay, the cells respond to light. Great. The next thing is, okay, not only do they respond, they also can to evoke a response in the visual cortex. Then the next thing they did was, okay, well, are the animals behaviorally responding to the light in the environment? So they did this kind of really simple, no behavior in mice is simple as a behaviorist, but they did this theoretically simple task where essentially you're just in a box. Half the box is light. A small portion of the box is dark. Mice will spend time in the dark. So in a normal animal, most of their time is going to be spent on the dark side of the box. So that theoretically, if they can't see, it's all dark to them. So what happens is when you lesion the retinas, essentially they spend more time in the light. And then what they show is they'll spend more time in the dark once they restore this kind of visual system function. The thing is, though, I guess when you're thinking about visual system and disorders, there's a really big difference between sensitivity to light and actual visual like
acuity,
seeing things. So just because an animal can respond to light, you're going to get and evoke behavioral response. You're going to get visual system responses, visual cortex responses. But the question is can they discriminate images? Can they actually see anything? And I mean, again, the first step, right? If someone can't see, just light responses is a great advance from nothing. But the ultimate goal is having these cells integrate appropriately into the visual cortex so that images can be made out, not just is it bright or not. And their kind of behavioral task is is there light or is there not light? And so I think that it doesn't kind of get at that, but at the same time, it shows that the animals are able to not only, evoke a response, but use that response to kind of make a decision based on how they're perceiving the environment. So, I mean, that is kind of the next.
One question I had about the behavior was that's a little bit strange. So the wild type mice that have normal vision are still only spending 50% of the time in the dark chamber. So there's a little bit. So usually mice are, I guess, when I think about mouse behavior, mice are just scared, right?
Yeah.
So they prefer to be in the dark where their predators can't see them. And generally, if you put in the light dark box, mice will prefer to be in the dark much more than in the light. And here they restore it to what they have as the wild type function, but there's something a bit strange about the fact that the wild type mice are only 50-50.
I agree. This is a weird way of doing this experiment anyway, where they're just plumping the mouse in the environment, and then they've made the dark chamber smaller as well, and I'm not quite sure.
I think the rationale for doing that is that then you don't have a spatial probability thing. So if it was 50% and it was all random, they should send 50% on each side. So then the argument is, it's smaller. But you do also have a problem in an open field like this with novelty induces motor activity. So you have a bunch of competing factors that can explain why they're spending more time in the light than the dark. But that's a problem, I think. So then, you know what I mean? In an open field, an animal will run around before they kind of habituate. So if you get that, then you're going to get more light time. But then it's also not answering your question, which is the problem, right? Because the question too is like, do they have differences in motor activity? Like what are these animals when they can't see, are they running around more? Are they running around less? Like what, is it deliberate? And I think I would have liked to see maybe a more complex analysis of even just like distance traveled. Right. Like, I think that kind of stuff would have been important for.
But, I guess one thing I would say is that I think if we're being reviewer 3 here and sort of asking for more and more experiments, but this in itself, I think having some functional readout is great. And, I guess the danger is if you throw some behavior into it, you're going to ask, well, if you did this bad behavior, why don't you do, better behavior?
Right.
On the other hand, the title alleviate symptoms of neurological disease in mice. So this is a big claim, and it's a little bit of a crazy scheme to do this. So I don't think it's unreasonable to ask for controls that, especially with the distance travel, which is data they should have from the experiment anyway.
Yeah, no, I totally agree. And I think actually, as Aaron probably gets more into this, the rest of the paper is pretty not that good. And so what they should have done is just concentrated on nailing this one paradigm from manipulation to behavior, rather than like try
another disorder. Yeah. I think that's the thing, right? Because the next thing they did, and it's kind of like going down the right path, is that they said, okay, well, let's look at the time course of how long it takes for these cells to kind of integrate into the visual system. So in figure 5, what they did is a whole time course of like how long it's taking for these cells that they're kind of differentiate into cells that are actually projecting back to the visual cortex and the superior colliculus and the lateral geniculate nucleus and the optic nerve. And so it's kind of cool because they show within a month that it's actually kind of integrated. So you can see kind of stepwise over time. They did it basically 1 week, 1.5 weeks, 2 weeks, 3 weeks, and a month, that it's taking a month, which isn't actually that long of a time for this to happen. So they did go into it. So I would have liked to see more of kind of this, like, why is this single gene in a glial cell enough to basically draw, like, is it just that? Is that started a cascade? Like, I would have liked to see, like, instead of going into the Parkinson stuff, what if they did sequencing after that knockdown to see what other gene programs come on at different time points to figure out, like, what is it that's causing this to first differentiate into a cell population that is not like glial cells? Then what is also, there has to be other things that are involved in actually sending projections to other brain regions. I mean, it can't just be this one protein and that's it. There has to be kind of some sort of cascade. And I would have liked to see a little bit more maybe about that, rather than kind of the next thing they did, which is kind of switch gears all together and go into a Parkinson's model with 6-hydroxydopamine lesions. But yeah, so essentially, and I think we can like go maybe into, because I think Jason's paper gets at the same stuff from the other half of this paper. But I think the really cool thing here, right, is that you have cells, you can lesion the retina, have cells be removed, and you literally can restore the visual response to light by using CRISPR to knock out this protein in glial cells. Like, that is really, really cool, right? You can basically use existing cells and have them differentiate into a new cell population that is responsive to light and also elicits responses in, the brain. Like, that's like the beginning of something really cool. And we know this kind of stuff can actually be used in humans because the eye is one place that we can actually access and use these kind of strategies to do this. So I think this is a kind of cool thing to say, okay, well, in disorders where you have loss of neurons, we're starting to figure out ways to actually regenerate them in a way that integrates into an existing system, not just puts them there, right? Because you can't just inject stem cells anywhere in the brain and it just integrates. So I think that's what I was so excited about this kind of this approach and this paper. Trying to think.
All right.
Yeah, I would say, do you want to go into kind of maybe, because I mean, I could go into other stuff, but I think it's kind of the same as what you're going to go into for this paper. So maybe we can go through yours, and I can bring up things that are parallel or.
Yeah, exactly. I think once we get to the dopamine stuff, I'll defer to you anyway.
I want to point out that's from a couple of groups in China, right? That
Right.
Yes.
Which, let me
Yes.
just.
I'm looking it up. Yeah, it's in Shanghai. So it's both of them are in Shanghai. Sorry, I'm really bad at doing this. So the Institute for Computational Biology at the Shanghai Institute of Nutrition and Health, and then the Institute of Biological Sciences at the University of Chinese Academy of Sciences.
Right.
So this is a number of groups in China.
Right.
Okay, so this second paper, which is in Nature, was published 24th of June 2020, and this is a few labs at UC San Diego, led by Jiangdong Fu and Don Cleveland. And so the premise of this paper is quite similar to the Cell paper in that they wanted to set out to see whether if they downregulate PTB in vivo in astrocytes, whether that would actually convert them into neurons, and if it does convert them into neurons, are those neurons functional in any way? And as Aaron said, one of the aspects of neurodegeneration is that particular disorders affect specific populations of neurons, preferentially early in the disease, and some more than others. So Parkinson's disease, which is the focus of this paper, of course, predominantly initially affects the dopamine-producing neurons in the substantia nigra. And so some of the context for this is that one idea was that if you knew which population of neurons you could restore, in effect, you should be able to reverse the phenotypes or be able to at least halt them. And so when stem cells came, and when we had the ability to make stem cells and to produce stem cells, there actually were quite a few clinical trials for Parkinson's disease where there was grafting of those stem cells into the brain with the hope that if you put them in the right place and maybe gave them some goodies, that they would actually regrow. And unfortunately, for the most part, those clinical studies were not very successful, partly because those stem cells actually didn't become neurons, or they didn't become the right neurons, or if they did become a neuron, they would not integrate into existing circuits. So the idea here is that, well, we have these astrocytes. Astrocytes are glia cells that can divide in the brain, and if they're in the right place at the right time and they have the right goodies, then maybe they can restore function if you make them into neurons. So similar to the other paper, they, instead of using CRISPR, then they actually use RNA interference, which is a little bit of an older method to downregulate expression of genes, and they use viruses to then express this RNA interference in vivo or in cultured cells. So in figure 1 here, they actually do talk a little bit about why this one gene can set off a cascade. And so it actually came, the original discovery was that there was this one transcription factor called REST that if you inhibited that transcription factor, you could convert astrocytes into neurons. And so the default state in an astrocyte is that there's a program that turns on this REST transcription factor, and that sets off this cascade of gene expression. And so if you inhibit PTB, that actually sets off a cascade that ultimately then reduces this REST protein, and so that's why you get this cascade. But what was interesting is that in the astrocytes, there seems to be a couple of latent programs. So it's almost like they're poised to become neurons, but there's just one doorstop, and if you remove the doorstop, then you open the door to those gene programs. And so that kind of explains, in a big level picture of why this one gene has such a dramatic effect. And they show that in both cultured astrocytes and mouse astrocytes and human astrocytes. And then they try to do this in vivo, where they use this viral approach to downregulate PTB, and they start off with both doing this in cortex of a mouse and also in the striatum. And so when they do that, they find that indeed after about 3 to 4 weeks, they start to see an upregulation of neuronal markers in the brain where they put the virus, and they can actually then also make slices from the brain to investigate the electrophysiological properties of those cells that are expressing the virus. So in their control virus, where they're expressing a scrambled RNAi construct, they of course don't see any changes in function. Astrocytes don't usually show any electrical activity per se, but when they do include this PTB RNAi construct, they actually start to see electrical activity, and by 8 weeks or so, they actually also start to see markers that in the striatum that suggest that they're becoming dopamine neurons, or dopamine-like neurons. So that's pretty cool in itself, right? So they're putting a virus that knocks down this one protein PTB in glia cells, and then those glia cells start to become neurons, and they start to show electrical activity that suggests that they are also actually integrating into circuits that are receiving action potentials and excited transmission. So that all of that sort of looks very promising. They also did some sequencing of those cells to sort of see what happens to gene expression. And as you'd expect, they kind of see a middle ground between pure astrocytes and pure neurons, but that over time, when they look at the AAVs that they injected in striatum, they actually see dopamine specific gene expression as well. So there's sort of some subtype expression. And I think actually one of the more intriguing aspects here was that when they compared both the sequencing and the functional output of these cells that they injected in the cortex versus striatum, they actually see the astrocytes that they converted to neurons in the cortex have slightly different properties than the astrocytes that they converted in the striatum. So one, it says that actually the gene program in the astrocytes is somewhat different in those two structures, and that even when you interfere with this pathway, there's some sort of local effect of on gene expression. So that, I think that's pretty cool. You guys interrupt if I'm sorry.
I already jumped ahead because I was like, oh, do these release dopamine? But I'm looking all the way to figure 5, and it looks like they actually did some of those studies. So I was okay.
Yeah. So again, that all looks promising. And so then they thought, okay, well, if we can reprogram these in glia cells and they're functional, and they look like they're becoming dopamine-like neurons, do they actually have dopamine-like functions as well? In figure 3, though, they did look also to see connectivity. So they, similar to the cell paper, they labeled axons, labeled projections from different structures in the brain to see if some of the connectivity looks normal. And again, when they downregulate the PTB, they do see retrograde. So they're able to look at the substantia nigra itself and to see if they put these retrograde beads in striatum that can go back to project back to the substantia nigra, and then they see that there are projections that are now there with the PTB, decreasing the PTB in substantia nigra. So that's yeah.
Let me just to take a step back on the anatomy. So the dopamine neurons are actually situated at a different part of the brain from where their axons project. And both in humans and in mice, these are fairly far distances, just like we talked about with the retina projecting into the brain towards the lateral geniculate nucleus. And one thing that they do here, which was different from the Cell paper, was that they convert astrocytes in the midbrain and then show, just like in the Cell paper, that they project into the striatum. So they're able to send axons along the kind of normal axon pathway. But what they also show is that if they inject the PTB shRNA into the striatum itself, they don't have dopaminergic cells they don't convert astrocytes into dopaminergic cells. And that's the opposite of what they show in the Cell paper, where they actually don't do any midbrain experiments. In the Cell paper, they only show conversion of astrocytes from the striatum into dopaminergic-like cells. So
Which
this is
strange.
Right.
That there's just dopamineurons, because in the striatums, these dopamineurons, they project into the striatum, but the cells in the striatum that receive the dopamine are not dopamine neurons themselves. There are other populations of neurons that integrate into other brain areas. So adding dopamine neurons to places where they don't belong is probably actually not a great strategy.
Well, some of the fetal grafts in Parkinson, so for a while there was this push in Parkinson's to inject fetal dopaminergic neurons into human striatum, and then they would release dopamine, and it would kind of be like a volume transmission, and that works. And the same with if you think about L-Dopa, this is you're just having this bath of dopamine in the striatum, so it could work. But I guess
how does it work forever? Doesn't it only work for a short amount of time? I mean, if you just increase L-Dopa forever, don't they become desensitized to it? Because you have receptor desensitization and all kinds of other downregulation of the existing dopamine terminals as well through autoregulation and things like that.
So I'm pretty sure the fetal grafts work for quite a long time. Okay. And in fact, there are some really interesting studies where they follow these patients out decades, or at 15 years, and they actually see that the fetal cells are still there. They still have symptomatic improvement, but some of them actually have alpha-synuclein deposits and show neurodegeneration. So that's been evidence for kind of transfer of these prion-like synuclein species. Yeah.
Okay. So back to the paper. But the cool thing here is, as you said, is that they only see these dopamine cells when in the midbrain. They don't see them in other parts of the brain when they do the same manipulation. So there's some unknown local factors, or maybe there's some intrinsic cell autonomous gene expression programs in the astrocytes that when you convert them in the right place, then they actually show these sort of differences in conversion. So that in itself, I think is pretty cool. So given all that, and they thought, okay, well, maybe if we are able to make dopamine neurons, can we use this to restore function in a Parkinson's model? And so here they use that 6-hydroxydopamine. Or is it
6-hydroxydopamine?
That's one.
Yeah.
Induced lesion. So basically, it's a chemical way of just killing those dopamine-producing cells in the substantia nigra, and it's been a model that's been used in rodents for a long time. And so they use that model to get rid of these dopamine cells, and then I guess one of the behavioral outputs of this is that they show different, and this is where again Aaron can chime in, but they show these sort of stereotypical movement disorder phenotypes that are kind of like Parkinson's disease. Like they go around in circles. You can induce them to only walk around in one direction, right?
Right. Yeah. So if you lesion one side, the animals can still move, but only in that kind of circular pattern. So it's actually kind of a very specific phenotype from this. So it's a easy behavioral output to, one, know your lesion worked, but two, know if you fixed it. So it's actually a kind of cool motor phenotype that's really obvious and simple, and there's not a lot of room for nuanced interpretation. Either they run in a circle or they don't.
Right.
So it's kind of cool.
So it's cool. So they see that these dopamine neurons do project to the striatum. So they did some of the connectivity. And then they did some of those retrograde labeling experiments where they could also just see if the connectivity is normal. So far so good. But then in this Parkinson's model, what they do is they in the lesion side, they inject their virus with the PTB, and they want to see then if that can, one, restore those dopamine neurons, and two, can it restore the behavior or the differences in behavior. And I'm not used to knowing. I don't really know how good these behavioral experiments are, but to me, what they do show is that in their lesion side, the rescue they're seeing, both in the amount of dopamine that they're that are producing and in the behavior, are pretty good, despite the fact that they're using a virus to replenish just this one population of neurons. So they do see functional recovery with this sort of one simple lesion model.
I mean, the beauty of these experiments is that they test the mice before doing the rescue, and then they watch the rescue actually happen. So these lesions can be variable because, as you imagine, you're injecting this toxin, you can have different amounts of denervation. But here they're doing the before and after in the same mouse, so it's really an elegant design.
How long does the regeneration take? Do you know?
That's an Ori question. I haven't worked as much with 6-hydroxydopamine stuff.
So the lesion. So the dopaminergic cells die within a couple days to a week, but usually you let the lesion sit for like 3 weeks to a month more to get like the circuitry to stabilize. And there's also a lot of mice will die from the lesion, and most mice that will survive are the ones like at 3 weeks. Very few mice will continue to die. So if you take all the mice 3 weeks after the lesion, then you have a good cohort.
Right, and I think I didn't see exactly how long they expressed the virus for, but it's, I think, at least 2 months or so. And then and the N's are low here. There's N03 and N04, but the actual functional recovery seems pretty dramatic. That's all good. But then I think one of the coolest experiments they did here was that there could be many reasons why you get functional recovery, and they wanted to directly show that the recovery was due to the actual neurons that were made from the glia. And so here they used our friend the DREAD, the DREADs from what we talked about last time, where they added a DREAD receptor to the AAV virus with the PTB RNAi. So in the same cells that express the PTB, that knockdown PTB, would also express the DREAD receptor. And so then in essence, they could then use this CNO drug to manipulate the activity of only the neurons that were originally the glia cells. And when they do that, they show that they can basically totally get rid of the rescue effect of these neurons by inhibiting the activity of that population of neurons. So, really kind of an elegant way of causally showing that this functional recovery is directly due to the glia cells that became neurons, not because of potentially other neurons integrating or taking over function or something. And so that I thought was a really cool causal experiment that you didn't see in the Cell paper. And then finally, in terms of translating this to humans, we talked about CRISPR as an approach for gene therapy, but there's still some issues there of getting CRISPR into human cells. And so one of the sort of more recent ways of manipulating gene expression or protein expression in humans is using these antisense oligonucleotides, which are these small DNA or RNA derived nucleotide sequences that you can that can bind to specific genes and inhibit their expression. And recently, there's been companies that have modified these ASOs so that they can be stable in vivo in a human. In fact, you can inject them, and so there's this new therapy for another neurodegenerative disorder SMA, which is which affects children, and in fact it's lethal, and there's no cure. And what they found is that if just one round of injection of these ASOs can inhibit the toxic gene or protein that's being made in these children. So it's been a real exciting development in RNA therapeutics. So anyway, so then they thought, well, if we could potentially knock down PTB using antisense oligos, this could then suggest a real pathway for therapy in humans. And so that's what they did. They injected these PTB ASOs into the brain of these mice, and they use some. It's true that they use some genetic ways of making sure that they could record how the astrocytes, whether the astrocytes become neurons or not. But essentially, it's a proof of concept experiment. So they put in these ASOs, and they do see a conversion of the astrocytes to neurons, and in they also then did some functional experiments in the Parkinson's model to see that they restore some function as well. And so that, so they in this one paper, they sort of go from mechanism to connectivity to functional output to behavior, and then to maybe even
to therapeutic relevance
using this in humans. So really pretty amazing piece of work, I have to say. I mean, it's really pretty amazing. And again, it's like it's a simple approach because it's just this one gene that they're manipulating. But I guess that there are going to be many questions about this. So we all sort of gloss over that astrocytes have function, but they do.
The long term. Well, that's my question is like how long term implications.
If you keep on converting astrocytes into neurons, at some point you're gonna have too many neurons and not enough astrocytes. So that may not be a good thing. But if it's an ASO, if it's a sort of transient effect that you can control, it does seem like once you convert these cells to neurons, they don't seem to revert back. It's sort of a trigger point. So it's exciting stuff.
So do they make the AAV vector specific for astrocytes, or is it
right? Yes, they do. So they use a GFAP promoter. So this is a promoter that's
only work in astrocytes. Okay. Right. By the way, you mentioned spinal muscular atrophy, right? There is a AAV carrying the spinal motor neuron 1 gene, which is used for gene therapy for these kids. It is the most expensive drug ever made. Treatment is $2.25 million.
Right.
Oh.
Yeah, and even these ASO treatments are hundreds of thousands of dollars per injection. So, not cheap.
What's the cost of a life? Yeah.
Yeah.
Well,
complicated.
If you can't afford it, right?
It doesn't matter, right? Yeah.
Yeah. I mean, I think insurance companies are. I mean, because the long term effect, the disability and the et cetera, et cetera.
Yeah, sure.
I think it's actually cheaper for insurance still, but it's it's crazy.
No, you're right. The cost to hospitalize and take care of these kids is a lot, and sure, but it's still a lot of money. And I just keep thinking of the very expensive CEO salaries that CEOs of every company are paid millions and millions of dollars, and why is that needed?
Right.
But I digress. Sorry. All right, that's great. Are you guys really excited about these developments? Is this someday going to happen in people, you think?
I'm always cautiously optimistic. I feel like when optogenetics came out, everyone was like, we're going to put them in human brains. And I think there's just this large jump between preclinical, this is neat, and think about this. So you do this, and kind of Jason brought this up, but the question is okay, maybe it works for the short term, but you have to monitor how long is this okay? Do these animals start showing symptoms of other neurological disorders for long periods of time? Sure. What are off-target effects? What are in these kind of things are, the first proof of principle is, can we fix a problem? And then the next thing is like, what other problems does this cause? Is it, how long does it last? I mean, when you're thinking about a lot of these require, I mean, the ASO approaches sound really cool, but a lot of the stuff people are thinking about require viral vectors.
Yeah.
Just expressing a viral genome in a cell may not be great for long periods of time. And in mice, we don't do these 10, 20-year studies, right? We say, oh, well, it's good for a couple months. So I think we still have a long way to go for refining these and making sure that they're not going to cause detrimental other effects. But this is a kind of exciting first step because there is, promise here, obviously.
Yep.
Yeah. And I mean, to be honest, I actually think this is, to me, these animal model sort of experiments, even in these initial papers, seem more promising than stem cells. And stem cells have been around for long enough now to say that they're not this magic cure. So I don't know. I think I'm definitely excited. I think that, and it's also in principle very sort of straightforward approach for therapy because it's just this one gene as opposed to sort of a cocktail of things that you have to manipulate. And I think also understanding why the astrocyte conversion seems to result in more functional neurons that are similar to the real deal, I think is really intriguing because it suggests that there's some sort of, I mean, we knew that astrocytes were in all these glia cells. There's subtypes of glia cells that are in different brain regions, but the fact that they sort of mimic or have these latent properties of the same neurons that are next to them is really, I think, intriguing.
Yeah.
Well, and I think the crazy thing too is it's like a one protein, one off, and then it seems like they stick around, which I mean, that has a lot of maybe treatment potential because you can think of ways where we can say, okay, let's get a vector or something that transiently expresses so that you don't have something there for a really long time. And then it integrates into the system. So it's supported now once it's there by the kind of micro environment. So you don't have to keep doing all this other stuff. I think that has way more potential than anything in the past, which is really, really cool.
And the ASOs are great. I think it just highlights this new therapeutic way to like specifically target individual proteins that we hadn't thought about yet.
Yeah, exactly. I mean, they are sort of injecting these straight into the brain, so it's sort of that's still another problem. But
yeah, but with these neurodegenerative disorders, I think this is a thing, right? Is it becomes really difficult to figure out when we try these approaches, right? Because like you think about things like substance use disorder, which I study, and it's really hard to justify like doing some in-brain injection because you say, oh well, this person, it's not, maybe they'll get better. So it's very hard. But with neurodegenerative disorders, a lot of times you're like, okay, well, there is kind of, it's going to keep degenerating. There is maybe some justification for trying these kinds of things. But, where you draw that line and how you do it, I think is what clinical populations are you going to target and where is it okay versus when is it not? I think that's the really, really hard thing with going through to the clinic. Or
that's true. I mean, in Parkinson's, they're already implanting electrodes into the brain.
Exactly.
So,
well, of course they use.
At Cornell, they're injecting viruses into instead of electrodes into PD patients. So there is a precedent for this.
Well,
well, especially when they're already doing surgical intervention. Sorry, Vincent.
It's okay. They also do inject polio viruses into gliomas, right? To try and, otherwise they die. Well, they die anyway. They get about a month. But yeah, so we'll see more of that, I'm sure. All right, we have one email before we wrap it up from Sue Ellen, who writes, "You read my letter last time on TWiN, and unfortunately you all thought I was being critical. I wasn't. I guess I'm cursed the way Vincent is. He's called grumpy even when he's not, and I'm called critical when I'm trying to flatter you all. I wrote because I thought you did an awesome job without Vincent. Yes, the sound quality could have been better, but I was interested enough in what he was saying that I was willing to go to my earbuds so I could hear better. And I even said in my email that I understood things are harder now because of the pandemic, etc. And yes, some of what is said on TWiN goes over my head, but that's true for all the Twix podcasts. Sometimes I listen twice or even 3 times if I think it will help. But then there are times when it's not going to help, so I just let it wash over me like an opera score.
I mean, if it makes you feel better, some of the stuff in these podcasts goes over my head as well, and I'm here discussing it. So,
Same. Yeah.
I like to say that sometimes Twix is like listening to opera. I know the basic storyline. I love the music, but usually I don't understand every word that's being said. Doesn't stop me from going to the opera. Won't stop me from continuing to listen to the Twix family of podcasts. So please don't think I was being critical because I wasn't. I was trying to say that you're all doing a great job. I look forward to every episode, and I'm learning a lot. Sue Ellen in Roswell, Georgia, where it's hot and humid, and COVID cases are up 40% since we ended the lockdown.
I think I actually like this feedback because I still don't actually know who's listening. So if anyone is listening and wants to like tell us, it's above their head or below their head or whatever, it's all great because then we can sort of figure out how to pitch the next few episodes.
Mm-hmm.
I mean, I know scientists, hopefully there's something for everyone in these podcasts, but it's good to know whether we're reaching the audience we're supposed to be.
Or no one at all. Yeah.
We're just talking to each other.
Yeah, right in.
I enjoy this.
Yeah.
But you can, if you want to write in, TWiN, TWiN at microbe.tv. And thanks, Sue Ellen. I didn't think you were being critical. It was fine. But thanks for writing, because I love the opera analogy. That's pretty cool. All right, TWiN at microbe.tv. If you like what we do, you can support us financially at microbe.tv/contribute. And you can find the links to papers and the letters at microbe.tv/TWiN. Today on TWiN, Ori Lieberman. He's at Ori Lieberman on Twitter. Thanks, Ori.
Thanks, Vincent. Good to talk to you guys.
Aaron Calipari is at the Aaron Calipari on Twitter. Thanks, Aaron.
Yeah, thanks, guys.
From Vanderbilt University. And, of course, Ori is at Columbia University. Vagelos College of Physicians and Surgeons. And Jason Shepard is at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Yeah, thanks, Vincent. And nice to chat with you guys again.
And I'm Vincent Racaniello. You can find me at virology.blog. Thanks for listening, everyone. We'll see you next time on This Week in Neuroscience.